This is going to be a recording of Chapter 14 for Baker College AP2 lecture. So first off, we're going to do a quick review on the characteristics of blood. And if you guys remember from AP1, we said that blood was the only kind of connective tissue that had a fluid matrix. And that fluid or the liquid matrix was the plasma. And inside that plasma, plasma was made mainly out of water, and there are other things that are dissolved in it and cells that are flowing in that water, and we'll take a look at those in a minute. That blood is responsible for transporting all of the vital substances to the different parts of the body. It's also important to maintain the interstitial fluid, and the interstitial fluid is the fluid that is found in between the cells. It distributes heat, so remember that the heart, because of all of the heart beating and the movement of the cardiac muscles, that was the main source of body heat. And with the blood flowing, it will distribute that heat to the different parts of the body. And and remember that about 8% of body weight is made out of blood. The adult blood volume is about 5 liters. That is an average, and it's obviously different from one person to another, depending on gender, depending on age, and whether the person is hydrated or dehydrated. But that again is just an average adult would be 5 liters of blood. For the blood cells, and we call those formed elements, those are mainly in the red bone marrow, and those are the red blood cells or erythrocytes, white blood cells or leukocytes, and platelets. You really need to be familiar with both names. And it says here that platelets are cell fragments, and you'll see where that name comes from. For the composition of blood, so this right here is all of your blood system. You can see here that 55% of it is the liquid plasma, where about 90% of plasma is made out of water. And in that water, you have different things circulating. For example, electrolytes, those are your salts, so sodium, chloride, potassium, magnesium. These are all examples of electrolytes. You also have proteins, and those proteins are albumins, globulins, and fibrinogen. Just a little intro on what the importance of these are. So albumins, that is responsible for what is known as the osmotic pressure of blood. And keep that in the back of your head, because when we talk about the kidney and how it works and the renal function and how urine is formed, albumins play a very important role in that. So you want to keep that in mind. Globulins are mainly the antibodies that circulate within our body, and those are obviously important for the immune system. So we'll talk about globulins again. We'll mention them when we talk about the immunity. And fibrinogen, fiber and gen. So gen means to generate or to create something, and fibrin, that's what we're creating. So fibrinogen is the precursor of fibrin, and fibrin is important to make blood clots, and we'll talk about that in today's chapter. Other things that you'll find circulating in plasma would be waste products, for example, carbon dioxide. Remember when we talked about anaerobic cellular respiration, and we said that sometimes lactic acid is formed, and that would be considered one of the waste products that our body makes. Another example of waste products would be a byproduct of protein catabolism, for instance, urea and uric acid. All of these are considered waste products, and they have to go out with the urine. We also have circulating in our plasma nutrients, glucose, for instance, different vitamins and hormones. And then last but not least, we also have gases circulating, for example, oxygen and carbon dioxide and nitrogen. So that was your plasma portion of blood. For the formed elements, and those are, as we said, platelets, red blood cells, and white blood cells, the majority, about 95% of your formed cells are actually red blood cells, followed by about, little bit less than 5% would be platelets, and then a very few would be white blood cells. Among the white blood cells, we've got 5 different kinds. 3 of them have granules, and we call those granulocytes. If the granules are neutral in color, meaning they're neither red or blue, they kind of have more of a purplish color to it, those are known as neutrophils. If the granules are red, they are known as eosinophils. And if the granules are blue, they're called basophils. Now, the monocytes and lymphocytes, these are two cells that do not have granules. Truth to be told is that they do have granules, but they are not, you can't see them under the microscope. So they're still considered agranulocytic cells, meaning A as in non-granulocytic. They do not have granules. So again, monocytes and lymphocytes, these are the two agranulocytic cells. And it's important to remember the names of these cells and the numbers. Well, not the numbers, but you need to know which one is, you need to know them in order. So we've got more neutrophils, followed by lymphocytes, followed by monocytes, followed by eosinophils, and last but not least, we've got basophils. So the mnemonic for the number of white blood cells would be nothing like me eating brownies. So neutrophils, lymphocytes, monocytes, eosinophils, and basophils. Again, that is just a mnemonic to remember the again, not the number. You don't have to know these percentages by heart, but you need to know which one is the most and which one is the least, and then you can go from least to from the cells with the most number or more percentage of cells to the ones of the least percentage of cells. Next, we're going to go to how are blood cells made. So again, the majority of our blood cells they're mostly made in the red bone marrow. They all start off as what is known as a hematopoietic stem cell. So it is a stem cell that we have as adults, and meaning hematopoietic. So again, poietic means to make something or synthesize something, and that something that we're making would be a heme cell or a blood cell. So all of our blood cells start off as hematopoietic stem cells, and then that stem cell has the ability to become one out of two different kinds of stem cells, either myeloid stem cell or a lymphoid stem cell. Lymphoid stem cells end up giving us only T or B lymphocytes. So you can tell from the name lymphoid stem cell that it'll give the lymphocytes, and we have two different kinds of lymphocytes. We've got, namely, there's T and B lymphocytes, while the myeloid stem cell will end up giving us all of the different kinds, the rest of the cells of blood. So it'll end up giving us blood erythrocytes or red blood cells, thrombocytes or platelets, and then all of the different kinds of white blood cells, except of course your T lymphocytes. For this image, to understand what this image is showing you, that all of these cells right here are the different stages that happen in your bone marrow, while the one at the very bottom of this page would be what shows up in your tissues, and then the part in the middle is what shows up in your bloodstream. You don't have to know the cells or the stages, or the how this happens in order, but you do have to be familiar with the different parts that we're going to be discussing right now. So again, your myeloid stem cells will give you all of your different cells except for your T and B lymphocytes. T and B lymphocytes come out of the lymphoid stem cell. For the pathway in order to get a red blood cell, the very last stage would be, before making the actual erythrocyte, would be a reticulocyte. And reticulocytes, if somebody does a blood smear, meaning getting a blood sample and taking a look at it under a microscope, reticulocytes are very minimal in number. If a person has a high reticulocytic count, that means that their body is making a lot of erythrocytes, or the erythrocytes are unable to mature from a reticulocyte to an actual mature erythrocyte, and that could be a sign that there's something unusual going on. For the platelets, which is this portion right here, Okay, you see that platelets come out of a huge mega cell, and that mega cell is known as a megakaryocyte. And platelets are just little fragments of that bigger cell. And that's why when we were talking in the very first slide, we said that platelets are fragments. They're not real cells. They don't have a nucleus. They're just fragments that are kind of pinched out of this megakaryocyte. And then for when we talk about the white blood cells, again, we already discussed the different granulocytes, neutrophilic, basophilic, and eosinophilic. And last but not least, I would like to discuss a little bit about the monocyte, and I apologize, I have no idea how to move this thing out of my way. But underneath here is an image of a monocyte, and then you see a monocyte and an arrow giving you a macrophage. So monocytes and macrophages are pretty much the same cell. If the cell is found inside of the blood vessel, it is considered a monocyte. If that monocyte leaves the blood vessel and goes into the tissue, it is now considered or called a macrophage. It changes a little bit and becomes a macrophage instead of a monocyte. So a lot of times you're going to hear monocytes and macrophages kind of used together. That is because, again, the monocyte is the precursor of the macrophage. The monocyte is the name of the cell when it's inside of the blood vessel. As soon as it leaves the blood vessel into the interstitial tissue, it is now called a macrophage. It is a highly phagocytic cell, both of them, monocytes and macrophages, means that one of their main function would be phagocytosis, and the main phagocytic cells that we have in our body are the M and M cells. So first M could stand for monocytes, then the N in the middle stands for neutrophils, and then the final M would stand for macrophage. And these are the three main phagocytic cells that we have in our system. Going to our lymphocytes, which are, again, both of them originate from the lymphoid stem cells. We've got the two T lymphocytes, and then we also have B lymphocytes. So T lymphocytes is responsible for what is known as the cellular immunity, meaning that these T lymphocytes are more aggressive and they attack foreign bodies and germs directly. Comparing that to B lymphocytes, these are a little bit more passive cells, and instead of attacking cells directly, they make plasma cells, and these plasma cells make antibodies, and it's the antibodies that attack the cells. And that kind of immunity is known as humoral immunity, and we'll talk about that more when we talk about the immune system. For origins of blood cells, and this is again, that image, the diagram, the flow diagram that we just talked about, that whole process of making blood cells is known as hematopoiesis, and they both originate, or all of them originate, in the red bone marrow from either hemocytoblastic cells or hematopoietic stem cells. So these are both the same kind of cells. And these hematopoietic cells can either give lymphoid or myeloid, or they can just start multiplying to give more stem cells. Okay? And as we said, that the lymphoid stem cells give rise to the both kinds of lymphocytes, T and B, while the myeloid cells give all the other different kinds of blood cells. So now we're going to talk about the characteristic of each cell individually. For the red blood cells, this is also called erythrocytes. Again, you have to be familiar with the different names. It kind of looks like a biconcave disc. This is another cell that does not have a nucleus. So the two cells in our bloodstream so far that we've talked about that do not have nuclei are our red blood cells and platelets. Red blood cells also don't have mitochondria, which means you would imagine that they would only produce ATP through glycolysis only. They cannot perform Krebs cycle or electron transport chain, because remember, those two happen in the mitochondria. In red blood cells, you'll find hemoglobin, and the color of the hemoglobin really depends on the absence or presence of oxygen. So if there's oxygen attached to it, that's known as oxyhemoglobin, and that gives the blood a little bit more of a reddish color. If there's no oxygen, that is known as deoxyhemoglobin, and that gives more like of a bluish color to it. And that's why arteries and veins, you'll always find them kind of color-coded, where the arteries are red because it has oxyhemoglobin, and the veins that usually carry deoxygenated blood would be kind of more the color-coded bluish in color. So here you can see an electron microscopic picture of erythrocytes, and you can kind of see them here being squeezed through the different capillaries. For the red blood cell count, the typical range is about, these are your ranges, okay, so on average it's about 5 million, depending again on gender and age of the person. So in males, they tend to have a higher count, while in adult males tend to be a little bit lower because of menstruation and the loss of red blood cells during menses. And you can see here again that kids, tend to have kind of be on the lower end. But again, on average for all ages, about 5 million per cubic, per microliter of blood, or cubic millimeter, which is a very, very small dot of blood. So you can imagine that the number of red blood cells is pretty high. Red blood cell count is one of the very first blood work that is usually asked for when any patient comes in. It can sometimes, it really cannot diagnose a disease, but it does give you kind of a general idea about the health of the patient. Okay, what it can do as well is that also evaluate for progress of disease. So if you find that somebody is diseased and they're anemic, and then you're trying to treat that chronic illness, and then you find that their red blood cell count is getting better, that that's what we mean by evaluating the progress of a disease. The amount of red blood cells we have, or the RBC count, is really a reflection of the ability of the blood to carry oxygen, and that's what is known as the oxygen carrying capacity of blood. So the more RBCs that patient has, obviously the better the capacity of that blood to carry oxygen. Now, in order to produce blood cells, that is known as erythropoiesis. So erythropoiesis, again, is the making or synthesis of red blood cells. In order to make red blood cells, there are chemoreceptors within our bloodstream, and those chemoreceptors are responsible for picking up how much oxygen is in our system. So if the chemoreceptors pick up low blood oxygen, that will stimulate the kidneys and liver to make a hormone known as erythropoietin, or EPO. Kidneys are the ones that are responsible for making that hormone, especially in adults. The liver has a little bit more of an important function in that. In utero, after delivery, both still make it, but again, kidney is more responsible for that function. So again, the kidney and liver, to a lesser extent the liver, will make the hormone erythropoietin, and that will circulate within the bloodstream and reach the red bone marrow and blood in the bones, and that will make the red bone marrow, or stimulate the bone marrow to make more RBCs, and then because those RBCs will be released into the system, that will increase the oxygen carrying capacity, and meaning there increase the blood oxygen back to normal, and that would inhibit this whole process from reoccurring. For the dietary factors that we need in order to make RBCs, we need a vitamin B12 and folic acid, and those are needed to make DNA synthesis in general, and that's why usually during pregnancy, a lot of women are prescribed, or most women really are prescribed folic acid, because that is necessary for the growth and the division of all of our cells, not just RBCs. But RBCs in addition also needs iron in order to make the hemoglobin. Hemoglobin is mainly has a lot of iron in it, and if the person isn't getting enough iron, that can lead to anemia. So we got different kinds of anemia. Anemia, if you look at the name, it is kind of a misnomer, meaning that anemia is not a correct term. So emia means blood and an means no. So anemia doesn't really mean that that person has no blood. It just means that the number of RBCs has decreased, or really a more specific definition would be the decrease in the oxygen carrying capacity of blood. And there are so many different reasons for it. The most common would be iron deficiency anemia, where the person doesn't have enough iron, so they're not making enough hemoglobin, and that person is unable to carry oxygen. Different kinds would be aplastic anemia, which means the inability of the red blood cells, of the red bone marrow, was destroyed and is currently unable to make RBCs, and that is usually because of radiation or toxic chemicals. You could imagine that in aplastic anemia, not only is the red bone marrow not making RBCs, it's also not making any white blood cells or platelets. Another cause would be a hemolytic anemia, where the red blood cells are being made, but they're being destroyed. So hemolysis means destruction of RBCs. We've already talked about iron deficiency anemia. Pernicious anemia would be a deficiency of vitamin B12, and that leads to defective RBCs where they're huge in size, and that's why it's also known as megaloblastic anemia, because the red blood cells are huge. This is it's known as a pernicious anemia because it's a very hard kind of anemia to treat. And again, you'll learn about this a lot more during in your pathology class. Sickle cell and thalassemia, these are both genetic kinds of anemia. Sickle cell disease is more common among African Americans, and thalassemia is more common among individuals that are originally from countries related to the Mediterranean Sea. So it's also known as the Mediterranean anemia. Again, you'll learn a lot more about these in pathology. Next, we'll talk about this red blood cell destruction. And in this case, we're not talking about a pathological destruction. We're just talking about the life cycle of a red blood cell. So I'm not going to really use this except for a couple of points here, that red blood cells on average, they have about 120 days to live. So you see that their lifespan is not a very long life. And they're usually destroyed because of all of the squeezing and the very small capillaries, and they tend to age and then die off. And they're going to be eaten up by macrophages that are found in the spleen and in the liver. Okay, so all the changes that we're going to talk about in this graph right here happens inside of the spleen and in the liver by the macrophages. So you see here that the RBC is made out of heme and globin. The globin portion is protein, and we all know that protein is the building blocks of protein are amino acids. So that globin protein will break down into its individual amino acids, and then the body can recycle those amino acids to make whatever it needs to do. While on the other side, the heme portion is made out of both iron and biliverdin. So if you look at the iron part has one out of two different routes to go. It could be recycled to make new hemoglobin, or it could be stored in the liver as ferritin. While the biliverdin, that is greenish in color, okay, and it will be excreted, and then it changes into bilirubin, which kind of has an orange tinge to it, and that will be excreted in bile. And that, these two chemicals is kind of what gives the bile its greenish color. So that bilirubin will be excreted in urine, but if the person is unable to excreted in bile, I apologize. If the bilirubin is unable to be excreted in bile, but now is kind of accumulating in the bloodstream, that will lead to jaundice. So you see now how that orange or yellowish discoloration leading to jaundice, why that happens. Here you can see the life cycle of a red blood cell. So we started off here in the intestine where all of the nutrients needed in order to make a red blood cell would be absorbed. So the folic acid, the vitamin B12, and the iron, all three of those nutrients will be absorbed and through the bloodstream will reach the red bone marrow, and that's where our red blood cells will be made. And then they'll do its function, and then 120 days later, they will be, by phagocytosis, the macrophages of the liver and the spleen. So the spleen is not in the picture, but it's just as important as the liver is. in this function. So so again, the macrophages will eat up the old RBCs, and old, that means again only 120 days old, and that hemoglobin will split it into a globin and heme. Globin again is a protein. It'll break down into amino acids and the body will recycle its use, while heme will break down into both iron and biliverdin. Iron can be recycled to make new RBCs, or it could be stored in the liver as ferritin, while biliverdin will change into bilirubin and then it'll be dumped into the bile. Again, both of these have, biliverdin is more like a greenish color, while bilirubin is more of a orange tinge to it, and that's kind of what gives the bile its greenish color. That bile is then dumped into the small intestine and it is passed out with feces. So you see that, we start off by absorbing the nutrients in the intestine, and then we end up throwing out whatever the waste is or whatever the bilirubin is down into the small intestine again. Now for the different kinds of white blood cells or leukocytes, their main function would be to protect against diseases. So leukocytes are also part of the immune system. We'll talk about them a lot when we talk about the immune system. They are produced in the red bone marrow, but the hormones that stimulate its synthesis are interleukins and colony-stimulating factors. So the hormone that stimulates the production of RBCs is erythropoietin, while the hormones that stimulate the release of leukocytes are interleukins and colony-stimulating factors. Again, we've got 5 different kinds of white blood cells. We categorize them into granulocytes and agranulocytes. Granulocytes are also short-living, meaning they have a very short lifespan, and those are neutrophils, eosinophils, and basophils. While the agranulocytes, like I mentioned, they do have granules, but you really cannot see them under the microscope, so they're pretty much non-noticeable, are your lymphocytes and monocytes, and these have a longer lifespan. Actually, some of the lymphocytes that we've developed because of our vaccinations or because of getting sick as kids, these can remain in our system for life. Function of white blood cells, all of them, okay, they produce their functions because of these little different things that they can do. So there's what is known as diapedesis, and diapedesis means, excuse me, that these white blood cells have the ability to squeeze themselves through the little slits that are found in capillary walls in order to exit the capillaries and reach the interstitial tissue. And that again is known as diapedesis, which is a function of our white blood cells. Phagocytosis, which is eating up any germs, foreign bodies, is a function of leukocytes, especially in, again, the main phagocytic cells of our body are M&Ms, or monocytes, neutrophils, and macrophages. Inflammatory response means that when they reach an area, some of our cells, and those namely our basophils, are able to release heparin and histamine. Heparin is a natural blood thinner, okay, that our own system, our body makes. Again, the basophils in particular make heparin. And the basophils also release histamine, and histamine would lead to vasodilation, and that leads to increase in the amount of blood coming into the area. So that helps with the inflammatory response and helps bring in more white blood cells to attack whatever foreign body is trying to get in there. And then positive chemotaxis means that white blood cells, when they reach an infected area, they produce different chemicals, and these chemicals attract more white blood cells to come in. It's kind of like they call in their other white blood cell colleagues to come in and help. Now to talk about these cells individually, neutrophils, these are your first responders. So whenever a foreign body the body realizes that there's something foreign within its system, the first one to come would be your neutrophils. So again, these are your first responders. They have a large multi-lobed nucleus, hence the name polymorphonuclear leukocytes. So if you break down that name, leukocyte is your white cell, right? Polymorphonuclear means, poly means a lot or many, morpho means shapes and shapes or shapes, and nuclear is just that, nucleus. So these are the white blood cells that have many shapes of their nuclei. And again, these are very strong phagocytic cells, and they're usually elevated in bacterial infection. So if somebody comes in and you order a white blood cell count, and you see that generally speaking their leukocytes are elevated, what you really want to do is break down which white blood cell is the one that is elevated. So you do what is known as a white blood cell count with a differential, or with diff, meaning that you want a breakdown of each of these white blood cells. So if you find that the neutrophils are elevated, that would be more indicative of a bacterial infection. So a couple of things that you want to keep in mind for neutrophils would be your polymorphonuclear shape of the nucleus, that they are the first responders, that they are highly phagocytic, and they elevated in bacterial infections. Next would be your eosinophils, and as you can see here, the granules tend to have a red color. The nuclei are bilobed, and these are mostly elevated in allergic reactions or parasites, like for worm infections, and elevated again in your parasitic worm infections and in allergies. Again, that is the importance of having a white blood cell with diff. For the numbers, you don't have to know the exact numbers, but again, you have to know them in order. Basophils would be the bluish granules, and basophils release histamine and heparin. Heparin, we already mentioned that it stops blood from clotting, and that means that in that little area where the inflammation is occurring, there is going to be, we've got a blood thinner to prevent clots from happening, so that more blood can come in and help fight that infection. While the histamine really stimulates the inflammation, it leads to vasodilation of the blood vessels, bringing more blood into the area. Monocytes, these are the cells that have huge kidney-shaped nuclei. Okay, and these are one of the phagocytic cells that we talked about. Remember, the monocytes, as we mentioned, when they leave the bloodstream, they become macrophages. Okay, and they again had agranulocytic cells, one of the agranulocytic cells. The other one we have are lymphocytes. And lymphocytes have a huge huge nucleus. We're just surrounded just by a little rim of cytoplasm. And lymphocytes are two different kinds. We already talked about those, the T and the B cells. And we said that T cells directly attack different germs or pathogens or tumor cells, while the B cells make plasma cells, and these plasma cells will make antibodies to do the work for them. Now we're talking about platelets, and platelets are also known as thrombocytes. And as we mentioned before, they're little cytoplasmic fragments of the huge megakaryocyte. They don't have a nucleus, just like RBCs, and the platelet count is around this range in within a microliter of blood. It helps. Platelets' function would be blood clotting, so stoppage of bleeding. And we'll see how that happens. Platelets in general release a chemical known as serotonin, and serotonin will lead to the spasm of smooth muscles, so it leads to vasoconstriction. Now for the steps of hemostasis. So again, make sure you're familiar with all of these different terms. Hemostasis, hematopoiesis, they all kind of sound kind of the same, so make sure you are familiar with all of them and their definitions. Hemostasis, that is the action or to prevent blood loss, and it's made out of 3 different stages. First off would be vascular spasm. Second would be your platelet plug formation, and then last would be blood coagulation, and they happen in this order. And these obviously are more effective. The smaller the blood vessel is obviously this, the easier it would be to stop bleeding. So first of these 3 steps would be vascular spasm. And we already mentioned how blood vessels constrict, it would be by these platelets releasing serotonin, and the serotonin leads to the constriction or the contraction of smooth muscles that are found within the blood vessels, and that will lead to vasoconstriction. And it's also stimulated by, as soon as the blood vessel wall is penetrated because of an injury, that could lead to pain, and these pain receptor reflexes also help in the constriction of the blood vessels. Although know that this response doesn't really last for a very long time. It only lasts for about half an hour, and then the effect of the serotonin is really pretty much non-existent. The second stage, or the second step in hemostasis, is the formation of a platelet plug. So you can see here, this is the injury or the break that happened in your blood vessel. And again, platelets are going to release serotonin, so leading to the vasoconstriction of the blood vessel. And then platelets are going to come and gather at that little part. So it's kind of like as long as the blood vessel wall is continuous, platelets are not going to attach, and you can see them just flowing within the bloodstream. But as soon as now the platelets are in touch with the connective tissue that is found in the wall of the blood vessel, you can see that they'll stick to that connective tissue, and they start sticking together, and you can see here that it's made a platelet plug. That platelet plug, though, is a very weak plug. As soon as, if any pressure occurs on it, that platelet plug will be dislodged and bleeding can happen all over again. So the body has to make something that is more secure, and that would be your blood coagulation, or an actual blood clot. And a blood clot is made of all of these little fibers. These fibers are made out of fibrin, and you can see it trapping a lot of blood cells. These are the red blood cells that are being trapped, and that is what a blood clot is. So a blood clot, again, is the fibrin fibers that have trapped a lot of red blood cells. In order to form a blood clot, there are lots of steps that are involved, and it's more of a cascade of events that happens. It's kind of like a domino effect. If you've stimulated the very first step of blood coagulation, you will find that all of these steps happen in order until eventually a blood clot is formed. And a lot of proteins are involved, and all of these proteins are known as clotting factors. Two different things that we need for blood coagulation or a blood clot to be formed would be vitamin K. Okay, we also need calcium. And the major event that happens in a blood clot would be the fibrinogen changing into fibrin and leading to the trapping of these cells. Now, there are two different pathways that can lead to a blood clot, and they are known as the extrinsic pathway and the intrinsic pathway. I'm going to go straight to the image in order to compare the two different pathways that we have. So on your left-hand side here, you'll find the pathway known as the extrinsic pathway, and then this is known, the one on your right-hand side of the image, is your intrinsic pathway. They both lead to what is known as your common pathway. You don't have to know the steps by, all of these steps, okay, but you have to be familiar with the parts that we're going to be discussing. For the common pathway, the most important part would be where prothrombin becomes activated by what is known as the prothrombin activator into thrombin. And then thrombin activates fibrinogen into fibrin, and fibrin will make your fibrin clot. So again, make sure you're very familiar with this common pathway, again, where prothrombin is activated into thrombin through the prothrombin activator, and thrombin will activate fibrinogen into fibrin, leading to a fibrin clot. Okay, so that again is your common pathway. So whether you start as an extrinsic mechanism or an intrinsic mechanism, you're pretty much going to end in doing the same fibrin clot. So let's talk about what are the differences between these two different clotting mechanisms. Well, the extrinsic clotting mechanism really depends on tissue damage. So if there's any tissue damage in the body, and now blood is released outside of your blood vessel, and it's now in contact with parts that it's never been in contact with. So it's in contact with, say, bone, for example, or skin. That will lead to the formation of a blood clot. Okay, so that again is your extrinsic clotting mechanism that is stimulated by tissue damage in the body, and that will lead to the activation of tissue thromboplastin. And then you see through a whole cascade of events that needs calcium, that it'll eventually go to your common pathway. While the intrinsic clotting mechanism is stimulated by blood contact with foreign surface on the inside of the body, for example, the connective tissue of the blood vessel, okay, that will lead to activation of Higman factor, and then through another cascade of events that again needs calcium, would lead to your common pathway eventually. So for your blood clotting mechanisms, make sure you're familiar with what activates each mechanism. Know that both of them need calcium. You also need vitamin K, and that your common pathway would be the activation of prothrombin into thrombin through the prothrombin activator, and then thrombin will activate fibrinogen into fibrin that will lead to a fibrin clot. Here you can see a summary of the 3 steps of hemostasis. So again, your vascular spasm, then your platelet plug formation, and your blood coagulation. Now for the fate of the blood clot, now that the blood clot is formed, what happens to that blood clot? Well, the body will start squeezing that clot so that the plasma is released from it. Kind of want to dry the clot. And through platelet-derived growth factors, these growth factors will stimulate the healing process, start stimulating the endothelial lining of the blood vessels to start healing, and then at the end, plasmin will digest these fibrin threads and dissolve the whole blood clot. And sometimes, unfortunately, thrombus is the formation actually of the blood clot, while embolus will be if a part of the clot is dislodged and then it travels through the bloodstream to a different part of the body. A thrombus is not a normal blood clot, it's an abnormal blood clot, meaning that a blood clot just happened on its own. Like, for example, DVT, or which stands for deep venous thrombosis, which would be blood clots that can happen in the lower limbs, which again, there was not preceded by an injury, it just happened. So again, that is a pathology, not a normal blood clot. Other different kinds of blood clots, so we already talked about thrombosis, we talked about embolism, and both of these can lead to infarction, which is gangrene or death of tissues because of the blocking of their blood vessels due to a blood clot. And atherosclerosis can also lead to the formation of a blood clot. So one of the things that our body does to prevent clots from happening would be the smoothness of the inside of the blood vessel. So here you can see a normal artery that has a very smooth endothelium. If you compare it to this blood vessel, you see here there's a lot of deposition of fat and accumulation of fat in the endothelium, leading to narrowing of the blood vessel, and it is now rough as opposed to this smooth lining, and that roughness can stimulate the formation of a blood clot. So how do we prevent blood clots from happening? Remember, we already talked about how some of our cells, for example, basophils, another cell known as mast cells, release heparin, which is a natural kind of blood thinner that our body makes. Just the smoothness of the lines of blood vessels prevent blood clots from happening. Again, if a blood clot starts to form, okay, the fibrin starts to latch onto different proteins, for example, thrombin, and then prevent the cascade from preventing the clot from spreading to other parts of the bloodstream. We also have antithrombin, which will help in inactivating thrombin if a blood clot is formed. So these two are really talking about the prevention of spread of the blood clot. So if a blood clot is starting to form already, but we want to prevent it from spreading, these are the two different systems that happen in the body. We've got antithrombin that kind of starts to deactivate thrombin, okay, preventing it from spreading the blood clot all over, while also fibrin can latch onto thrombin and prevent from the spread as well. This is a table showing the different cell components of blood. I will let you read that. Then we're going to talk about blood groups and transfusions. And these are important things to understand for both your lecture and for lab as well. So in about 1910, there was a whole bunch of antigens, genes that were discovered, and these are known as the ABO blood antigens. There are about 31 different kinds of genes that determine what our blood types are. Okay, and a little bit about some of the terms that we're going to be using would be antigen and antibodies and agglutination. So antigen and antibodies, they're both different proteins. Antigen, though, is in the case of, when we're talking about blood groups, an antigen would be a protein that is found on the surface of a red blood cell. Okay, antigen, more generally speaking, gen means to generate, anti means to generate the immune system or to generate an immune response because of the presence of this protein. But if we're talking about the antigen within the ABO system or blood grouping, that would be a protein that's found on the surface of a red blood cell. While an antibody is another protein, and that is found circulating within your plasma, in this bloodstream. So an antigen, again, in their ABO system is found on the surface of a red blood cell, while the antibodies are the different proteins circulating in your plasma. While agglutination would be clumping of the red blood cells. So if the antigen and antibodies are the same, so if you have an antigen A and an antibody A, that would mean that this person received the wrong kind of blood transfusion. That will lead to clumping or agglutination of these RBCs. Okay, we've got about 33 different kinds of antigens on the surface of your RBCs, but only a couple of them are really important for blood transfusion, and those are antigen A, antigen B, and antigen D. So we'll talk about these different antigens and what kinds do we have. We've got antigen, the different kinds of blood types that we have blood type A, B, and AB or O. So your blood type, the name of the blood type, depends on the kind of antigen you have on the surface of your RBC. So if you have an antigen A, that means your blood type is A. If you've got an antigen B on your red blood cell surface, that means your blood type is B. If you've got both antigens, then you've got your blood type is AB. If you have no antigens at all, then your blood type is O. Now for the antibody, remember you don't want the same kind of antibody in your bloodstream because then you will be destroying your own RBCs. So if your antigen A is present, then you would have antibody B circulating in your plasma. If your blood type is B, meaning you have antigen B on your surface of your RBCs, that would mean you've got antibody A. For blood type A, they have both antigens, so they have no antibodies at all. For blood type O, they have no antigens, and that means they've got both antibodies. This is just the blood type frequencies in the US. As you can see that generally speaking, blood type O is the most common, and that is also known as the universal donor. So anybody that has a blood type O can give blood to any of the other kinds of blood types. While type AB is known as the universal recipient, they can receive blood from all of the other blood types. So blood type O, again, is your universal donor, while type AB is the universal recipient. And here you can see a better demonstration of the antigen and antibodies. Right here you've got your RBCs with the little antigens on them, and then these are your antibodies that are circulating within the bloodstream. This individual has an antigen A on the surface and antibody B, so that person is type A. And you can see here the person here, this red blood cell has antigen B, that person is a type B blood. On this, you don't see any antibodies at all, and it has both antigens, A and B. This individual has type AB. While on here, you can see no antigens on the surface, but you see both different kinds of antibodies circulating, so this individual is type O. Agglutination, again, would be clumping of the RBCs. So let's say somebody, in this case, that person has, this is a blood type A person because the antigens are A. Let's say they got the wrong kind of blood transfused into them. So now that these anti-A antibodies are going to start attacking the antigen A that's on the surface of this red blood cell, and that'll lead to clumping or agglutination of the red blood cells. So basically the antibodies are going to destroy the RBCs, and that is known as an incompatible blood transfusion, which can be fatal. So for compatible blood transfusions, again, you want to make sure that you've got the right blood type. If you can't find the right blood type, or you're kind of don't have the time, your safest bet would be to go with the universal donor, or type O. Again, AB is a universal recipient. They can take blood from any other type, but they cannot donate. They can't give blood except to another AB person. Oops. Here, and we'll talk about this in lab as well, how to figure out what the different kinds of blood types. Here you got a type, this right here is your anti-A serum, and that's your anti-B serum. And you'll put a dot of blood, on different slides, and on top of each dot of blood, you're going to put a little bit of that anti-serum. And then you want to figure out where the agglutination happened. So you can see here, this is where the agglutination happened. So this person has, because it happened with the anti-A serum, this individual had the antigen A on top of its RBCs that were attacked by the anti-A's, and that's why this individual had blood type A. If you look at this one, the agglutination happened with anti-B, so this individual has blood type B. If the agglutination happens in both, that person is blood type AB. If there's no agglutination, like the one on the bottom, that individual is blood type O. And then we also find that sometimes we say, or not sometimes, always we say, blood type O positive or blood type O negative. So what does the positive and negative stand for? They stand for another antigen known as the rhesus factor, or the RH factor, also known as antigen D. Okay, so the RH antigen or antigen D are the same antigen. RH stands for rhesus. Rhesus monkeys, because this antigen was first discovered in monkeys, in the rhesus monkeys, and then so it was named after them, and then we found out that we have the same antigen, so we kind of kept the name. It's now commonly known as the RH factor or the D factor. So if somebody's Rh positive, that means that the person has antigen D or the Rh antigens on their RBCs. An individual who's Rh negative does not have these antigens on their RBCs. So when we give somebody a blood transfusion, if that individual is an Rh negative, we want to make sure that the blood they get is Rh negative as well. Because if they get an Rh positive, that means their body is exposed to this Rh antigen for the very first time. It'll start making antibodies, and can start destroying those red blood cells. And that is really an important thing to keep in mind, especially during pregnancy, and because it can lead to the destruction of the red blood cells of a fetus, and this is known as erythroblastosis fetalis, or the hemolytic disease of the newborn. So fetalis means fetus, erythroblastosis, osis means a condition, blast means to destroy, and erythro means the red blood cells. So we'll see what happens here in Rh incompatibility. So what do we mean by Rh incompatibility? This is an issue if the mother is Rh negative and the fetus is Rh positive. And the only way that can happen would be that if the father of this fetus is Rh positive. Okay, so again, your problem would be if there's a mom that's Rh negative, but the baby is Rh positive. That means that this fetus has an antigen that the mother has never seen before, and that again is your Rh antigen. During delivery, there is usually a little bit of mix of blood between in the placenta between mom and baby. So now you can find here that some of the antigen has reached the bloodstream of mom. She's never seen that antigen before, so her immune system is going to make antibodies against it. This first baby that was born is fine. There's no harm to that first baby. The harm comes that if this Rh negative mom becomes pregnant with another Rh positive fetus, now mom has antibodies against the Rh antigen, and these antibodies, as you can see here, will attack the blood of the fetus. It will attack the RBCs of the fetus, leading to the destruction of the fetal red blood cells. And we can imagine that that is dangerous condition for baby. And that again is the neonatal disease known as the hemolytic disease of the newborn, or erythroblastosis fetalis. This concludes what we need to know for Chapter 14. If you have any questions, please let me know.